Scientists knew that DNA stores genetic information and that proteins perform most of the work inside cells, but for a long time, one question remained unanswered. How does information stored in DNA eventually become a protein? In 1958, Francis Crick proposed an idea to explain this flow of information. He called it the central dogma of molecular biology. The word central was used because this idea describes a fundamental principle of how living cells work. The word dogma was Crick's own choice. The central dogma describes the flow of genetic instructions inside a cell. In its simplest form, it can be written as this. But this does not mean that DNA physically turns into RNA, or that RNA physically turns into a protein. Instead, the information stored in DNA is transferred step by step. The first step is called transcription. During transcription, the information in a gene is copied from DNA into a molecule called messenger RNA, or mRNA. You can think of mRNA as a working copy of the instructions stored in DNA. Once this RNA copy has been produced, it can be used to build a protein. And that's the second step, called translation. During translation, ribosomes read the instructions carried by mRNA and use them to join amino acids together in the correct order. This produces a protein, which is incredibly important as they build cellular structures, speed up chemical reactions, transport molecules, and carry out most of the essential functions for life. The central dogma also explains why genes influence our traits. A gene contains instructions for making a specific protein, and those proteins help determine how cells and organisms function. Scientists later discovered a few exceptions to the simple DNA to RNA to protein pathway. For example, some viruses can copy RNA back into DNA through a process called reverse transcription. Despite these discoveries, the core principle remains the same. The instructions stored in DNA are passed to RNA and ultimately used to build proteins. This flow of information forms the foundation of modern molecular biology.